Thank you, sir. Yeah. Hello. Good evening, everyone. I'm extremely sorry for the delay. Actually, I was facing some technical issues. Uh, I hope I'm audible and visible to everyone. Okay. So I will start. Yes, yes. The screen is visible to all, right? Okay. Okay, so yeah, good evening everyone. So I'm Akash Jyoti Pathak. I am one of the PMRF TA for this course, Molecular Biology, which is being run by Professor Vishal Trivedi. He's a professor at IIT Guwahati. So yeah, I'm a PhD scholar in IIT Bombay, and I'm working in NMR spectroscopy lab here at IIT Bombay. So I hope everyone has gone through the week one lectures. So just a second. So, can anyone summarize what all have been covered in week one? Anyone? Anyone would like to summarize what has been covered in week one? Okay. So in week one, we started discussing about the prokaryotic cell basics of. We have started the basics of uh, biology, we can say, because we started with prokaryotic cell and eukaryotic cell. How what are the structures that are present in a prokaryotic cell, um, like the cell wall of a bacteria, then how is the genome of a bacteria, what is plasmid, all these we had seen in lecture one. And then in lecture one, we also saw a demo how we can isolate a plasmid in a laboratory using the alkaline lysis method, right? And then in the lecture two, we saw the structure of an eukaryotic cell. So what are the different organelles, uh, starting from the nucleus and the vesicular B cell organelles, mitochondria, cytosol, chloroplast, then organelles for vesicular trafficking. And then in lecture three or four, there was a discussion on culture media preparation and also how mammalian cell culture media is prepared in laboratory, how it is filtered using zero point 0.2 micrometer filter paper, right? And then towards the end, we had looked into how cell fractionation is done. So, if you want to fractionate the components in a cell, so how we can do that? So, there are two methods. One is differential centrifugation, and another is density gradient centrifugation, right? So, I hope these you can recall whatever you have after going through these lectures. This is just a summary of whatever we had covered in week one. So if there are any specific doubt regarding any particular topic which you couldn't understand, then I can discuss that, or else I can start with the slides that I have prepared for this tutorial. Okay. So yeah, if anyone is facing any doubt or any issue, then you can just unmute yourself and tell me if you are not able to view the screen, or if my voice is not audible. Just a minute. uh, Someone wants to join again. Okay. Okay, so who can tell what is a prokaryotic cell? So we have been learning about prokaryotic cell from class 9, right? 7 or 8 we have been learning about prokaryotic cell. So the prokaryotic cell is basically, if you want to understand in very, they are bacteria. Bacteria mainly comes under the prokaryotic cell, and they have, yeah, yeah. Anyone wants to say something? So as this, yeah. So as this name, so as the name suggests, prokaryotic, pro means what? Pro means the word pro means primitive, right? And carry on means what? It means nucleus. So primitive nucleus, or we can say the organisms which are having primitive type of nucleus, or they are mostly there are these archaebacteria, and bacteria comes under this prokaryotic cell. So they have very simple structure, and they are very small in size, right? They are small size. uh, they have a very small and simple structure, and mainly bacteria. If we talk about the two members of these classes, one is the archaebacteria and another is the eubacteria. So archaebacteria, they can live in extreme environment, while bacteria, they can live in a wide range of environment, right? So they have a simple and small structure. If you look at the DNA, the DNA has around zero point 0.6 million to 5 million base pairs, and their DNA is, they have circular DNA, right? The DNA of prokaryotic cell, it is not linear. They have circular DNA, and also this DNA. This DNA remains freely organized in the cytosol, right? So it is not present in any organelle like the nucleus, which is present in the eukaryotes. So that is why it is not bounded by any nuclear membrane. So that is why this is called primitive type of nucleus, and that is why this structure, it is called nucleoid. Nucleoid means what? It means nucleus like, right? So that is why it is nucleoid, this structure. And then they have the cell wall, plasma membrane, and ribosome. So here on the right side, you can see the structure of a prokaryotic cell. So the outer covering, that is what? That is capsule. So capsule is a structure which is present even outside the cell wall, and this capsule provides the antigenic property to the bacteria, or pathogenicity of the bacteria arises because of this capsule covering, which is present in bacteria. Then inner to the capsule you have the cell wall, and inner to the cell wall you have the plasma membrane. And then if we look further inside, then there is the cytosol, and these blue colored dot structures that you see, those are the ribosomes. What type of ribosomes are present in prokaryotes? They are the 70s ribosomes, right? And then the bacteria, they have flagella, pilus. So these all things, if you must have read even in class 11th and 12th. So, and then coming to eukaryotic cells. So eukaryotic cell, they are larger in structure compared to prokaryotic cell. And eukaryotic means what? If you look at this term, eu means true and karyon means nucleus. So true nucleus. And so if you look at their structure, they have a much complex structure. So here on the left side is the structure of an animal cell, and on the right side is the structure of a plant cell. So you see there are one of the main differences between prokaryotic cell and eukaryotic cell is that the eukaryotic cells have membrane bound organelles. So you see there are different types of organelles which are present inside the cell. So the starting from this mitochondria, peroxisome, centrioles, Golgi complex, endoplasmic reticulum, nucleus. And if you look at the nucleus, this nucleus is surrounded by a nuclear membrane, right? And the nuclear membrane have pores which are called the nuclear pore complex. So we will look at each of these organelles in more detail, right? And then so this is the plant cell. So compared to the animal cell, if you compare the you can easily differentiate between the animal cell and plant cell. You see in a plant cell the vacuole is much larger, and then there are structures called chloroplast which are lacking in animal cell. Plant cell have a cell wall. So these are very basic things, right? You all have read this in your class 9, 10th. Okay. So yeah, so okay, so if we see the differences between a prokaryotic cell and an eukaryotic cell, then nucleus. So prokaryotes don't have a nucleus; they have a structure which is called nuclei. So nucleus, we will only tell nucleus because if it has a proper nuclear membrane, then nuclear pore, nucleoplasm, all these if they are present, then only that structure will be called as nucleus. But prokaryotes do not have such structure. The DNA or the genome of a bacteria, it is loosely arranged in the not loosely, but it is freely lying on in the cytoplasm, right? Uh, yeah. So anyone has any doubt? So yeah, nucleus absent, eukaryotes in eukaryotes. I will just go through this very quickly because you all know about these. Diameter of typical cell is 1 micrometer. Eukaryotes have a larger. The size of the cell is larger, 10 to 100 micrometers. Cytoplasmic organelles are lacking in prokaryotes. They are present in eukaryotes. The DNA content of a prokaryote is around what? Of a prokaryote is around 1 million, while of a eukaryote is around 6.6 billion, right? Or around 1.5 into the, so that is the uh, the size of a human genome, right? 3.3 billion base pairs, right? And the chromosomes, if we look, the prokaryotes have single circular DNA molecule, eukaryotes have multiple linear DNA molecules. Okay, so you can go through this. I will share these slides. You can read about all these. So, what are the features of eukaryotic cell not found in prokaryotes? So, nucleus is present, cytoplasm separated by a nuclear envelope, right? All these points you all know. In plant cells, cellulose containing cell wall is present. Okay. Okay. So, yeah, the features of eukaryotic cell not found in prokaryotes. First of all, their division of cells into nucleus and cytoplasm. Um takes place in eukaryotes, right? So karyokinesis, cytokinesis, these all take place in eukaryotes. Then the cytoplasm, they're separated by a nuclear envelope containing complex pore structures, and their complex chromosomes are present. The chromosomes which are made up of DNA and associated protein, different cytoplasmic organelles are present, specialized cytoplasmic organelles for aerobic respiration, mitochondria, photosynthesis, these are present. Okay, then complex cytoskeletal system is present, flagella and cilia. So you can go through this. Okay. Now coming to the bacterial cell wall. So if we look at the structure of the bacterial cell wall, why is it important to study the bacterial cell wall? Because based on the cell wall of bacteria, bacteria can be classified into two groups. One is the gram-positive bacteria, and another is the gram-negative bacteria. Okay, so the cell wall of bacteria, you know, it is composed of a it is made up of a structure which is called peptidoglycan, right? So peptidoglycan is a polymer basically. It is a polymer which is composed of, yeah, it is a peptidoglycan is a polymer which is formed by repeating units of the of N-acetyl glucosamine and N-acetyl muramic acid. So this figure I've taken from the book Prescott. You see these are the repeating units of NAG and NAM. They are repeating, and NAM, the NAM molecule is attached to a tetrapeptide structure, right? So you see here. So this is the structure of NAG. Just So this is the structure. This is a sugar derivative, right? N-acetyl glucosamine. This is the structure of NAG, and the NAG and NAM is bonded to each other by a glycosidic bond, right? What is this glycosidic bond called? Anyone? So this is beta 1,4 glycosidic bond, which is present between the NAG subunit and the NAM subunit, or the NAG monomer and NAM monomer. And the NAM monomer is further attached to a tetrapeptide. So this tetrapeptide, you need to remember the name of these four amino acids. L-alanine, D-glutamic acid, mesodiaminopimelic acid, and D-alanine. So these four amino acids remain bonded to this the NAM monomer or the NAM sugar unit. Okay. So yeah, so the bacterial cell wall, it lies outside the plasma membrane. It is composed of peptidoglycan. Peptidoglycan polymer has repeating units of NAG and NAM sugar derivatives. And what is the bond which is present between the NAG and NAM sugar derivatives? It is beta 1,4 glycosidic bond. And the NAM monomer is attached to a tetrapeptide side chains with alternating D and L amino acids. Okay. So the humans have D amino acids. Are D amino acids present in humans? Proteins of humans? Yeah. It is not present. It is only present in bacteria. Okay. So this D type of amino acids you can see in bacterial cell wall. Okay. So till now everything is clear, right? And also one more thing is if you look at the structure of peptidoglycan in more detail, you will see that uh, other than the one NAM subunit, it remains to attached to another NAM subunit by a glycine. There are multiple repeating units of glycine bridge which connects one NAM unit to another NAM unit, and this forms a mesh-like structure which which provides the structure of the cell wall. So these rod-like structures you can see, right? These gray colored. So these are the glycine, pentaglycine interbridge. So these are the pentaglycine interbridge which connects one NAM unit to the other NAM unit and helps to form give the rigidity to this structure. Okay. So now talking about the so what is first of all what is a gram positive and gram negative bacteria? Anyone? Okay. And what is the basis of calling a bacteria gram positive and a bacteria as gram negative? So based on whether a bacteria can take up the gram stain. If a bacteria takes up the gram stain, then we call it as gram positive. And if a bacteria cannot take up the gram stain or it gets washed away, I think many of you have done this gram staining experiment in your practicals. So if a bacteria takes up the gram stain, then it is called gram positive. And if this crystal violet dye that is used for gram staining, if it gets washed off during the decolorizer or the decolorizing step, then we call it gram negative bacteria. Okay, so let's look at the structure of this gram positive and gram negative bacteria, and then we will learn about the gram staining method. Okay, so this gram positive bacteria have a thick peptidoglycan layer. Yeah, as someone was mentioning right now, yeah, you are correct. They have a thick peptidoglycan layer, and with this peptidoglycan layer is just present outside of the plasma membrane, right? And between this peptidoglycan and plasma membrane, there is a space, and this space is called the periplasmic space. Okay, another striking or another thing to remember about the cell wall of gram positive bacteria is they have this lipoticoic acids teichoic acids which are present in this peptidoglycan or which remains in the cell wall. The teichoic acid, this structures are present in the peptidoglycan layer, and these structures are absent in the gram-negative bacteria. Okay, so on the right, this is the structure of gram-negative bacteria. So you see there are three different layers in a gram-negative bacteria. First, you have the plasma membrane, right? The plasma membrane, which is composed of lipid bilayer, and then outside the plasma membrane there is this peptidoglycan layer. The peptidoglycan layer in this gram negative bacteria, as you can see in the figure, is very thin compared to a gram positive bacteria, and outer to this peptidoglycan layer is the outer membrane. So there is there is another membrane which is present outside the outer to the peptidoglycan layer, which is called the outer membrane, and this outer membrane has many channels. These channel proteins, these are called the porins, and also this outer membrane has lipopolysaccharide. Lipopolysaccharides, these structures are absent in gram-positive bacteria. Okay, so the gram-negative bacteria have an outer membrane, and this outer membrane is composed of lipopolysaccharides, right? And the peptidoglycan layer is very thin in gram-negative bacteria. So these are the, and the teichoic acid, which is absent in gram-negative bacteria. Okay, so let's look at the differences now. So yeah, so this is another figure. This is again from this Prescott book. I have taken this figure, and so the gram-positive bacteria, that stains purple, right? And it stains purple, and it retains the stain. The stain during the gram staining procedure, it retains the stain. Uh, they have thick cell wall composed primarily of peptidoglycan. The teichoic acid is present in gram-positive bacteria, and they have smaller periplasmic space. They have smaller periplasmic space because the peptidoglycan layer is very thick. And now, if you see the structure of a gram-negative bacteria, you see here. So first, there is this plasma membrane. Okay, so there is the this plasma membrane inner to the or outer to the plasma membrane. There is the thin peptidoglycan layer. Then there is another periplasmic space, and then again the outside the peptidoglycan layer, there is this outer membrane. Okay, so plasma membrane, peptidoglycan, and then there is an outer membrane composed of lipopolysaccharide. So it stains pink or red when you counter stain it with safranin, right? It stains pink, and they have a thin peptidoglycan layer, outer membrane present outside the peptidoglycan layer, outer membrane composed of lipopolysaccharides, and they have larger periplasmic space because the peptidoglycan layer is very thin in gram-negative bacteria. Okay. So now tell me, who can answer this question? Peptidoglycan is a polymer of. Uh, okay, yes. So everyone agrees with option A. So we discussed just now that it is made up of repeating units of sugar derivatives NAM and NAG, N-acetyl muramic acid and N-acetyl glucosamine. Okay, and also try to remember the bond which is present between these two sugar derivatives. That is beta 1,4 glycosidic bond. Okay. Okay. Now, yeah, so yeah, what type of linkage is present in peptidoglycan layer? That is beta 1,4. Okay. So now, this is one of the question which was there in practice week zero. In week zero, this was one of the question. The main reason that gram-negative bacteria lose their crystal violet dye during the decolorizing step is. So, can anyone briefly tell about the gram staining method? What are the steps which are followed in gram staining? So, if yeah, please go ahead. Yeah, yeah, yes. So, what is the decolorizer? What component is present in that? It's alcohol. Yeah, right, right. Safrine. Okay, so the decolorizing step is that when you dip this slide into this ethanol ethanol solution. Okay, so in this decolorizing step, what happens is that the gram negative bacteria lose their stain, while the gram positive bacteria retain the stain. Okay, so yeah, tell me the answer of this question now. So the first option is they have a thicker option A. Okay, anyone else? Any other answer? Option C, option D, which one? The correct answer is option C. Actually, the correct answer is option C. They have an outer membrane containing lipopolysaccharides that are dissolved by the decolorizer. Okay. Uh, the peptidoglycan, thicker peptidoglycan is not actually the answer, because the peptidoglycan, if you see the pore of the peptidoglycan layer, it will be same in both gram positive and gram negative bacteria, right? So, on the basis of the thickness does not control whether it can trap the, or whether it can retain the stain. The answer is that, so in during this decolorizing step, the this outer membrane containing lipopolysaccharide, that gets dissolved, and because it gets dissolved, the this violet color or the blue stain is lost. Okay, so yeah, you can see the solution. So the basic principle of gram staining involves the ability of the bacterial cell wall to retain the crystal violet dye during solvent treatment. So initially, all the bacteria take up crystal violet dye, right? So as someone was mentioning, that we first make a smear of the bacterial or the culture, and then then we stain it with crystal violet. So during this staining procedure, both the gram positive and gram negative bacteria, both of these bacteria will take up the crystal violet dye. However, with the use of solvent, the lipid layer from the gram-negative organisms is dissolved. With the dissolution of the lipid layer, the gram-negative loses the primary stain. Okay. So once this, because the this lipopolysaccharide layer or the outer membrane that is present outer to the peptidoglycan layer, right, in gram-negative bacteria. So when you dip your slide or this glass slide containing the smear into this alcohol, then once this lipopolysaccharide layer gets dissolved, along with it the dye also gets washed away. Okay. So that is why, because of the loss of this layer, these gram-negative bacteria uh, lose the blue color, and then you have to again counter stain it with safranin. Okay. Yeah. So I have shown this is figure, step one, step two, step three, step four. So yeah, I will move forward. So yeah, yeah, please. Uh, Primary stain with secondary stain. So your primary stain is crystal violet, right? And your secondary stain is safranin, right? Uh, so uh, so if you stain it with so if you use safranin as the primary stain, uh, then uh, then what is this property of the safranin? What type of dye is it? Do you know? Acidic dye, basic dye, basic, right? And crystal violet is also a basic dye, right? Uh, I remember this concept actually, but don't remember the detail actually. I think both of these are basic dyes. So if you stain it with safranin, if you take the primary stain as your safranin, then initially both of these bacteria will take up the stain, right? And uh, once you do this decolorizing, in this decolorizing step, these gram positive bacteria, they will retain the safranin dye, and while a gram negative bacteria, they will lose their safranin dye. I think that is what will happen. I have to I will confirm this question actually. I had read about this much earlier. I think I will confirm the answer in the next class, but that is what I think. Anyone from the audience, do you know answer? Do you know the answer to this question? Yeah, because yeah, please. Yeah, yeah, that is actually the, yeah, yeah, that is true. Yeah, you are correct. Okay, okay. Mhm. Yeah. Mhm. Yes, yes, you are correct. Yeah, you are correct. Actually, yeah, that is the correct answer. Both will take up the die, but the problem will be even the gram, because of the intensity, intense, the as she mentioned just now, that it is quite intense. So you will not able to differentiate between the gram positive and gram negative because. No. Mhm. Yeah, yeah, yeah. Yes, yes, yes. You are correct. Yes, she is correct. Uh, Yeah, because both of these, though both crystal violet and saffronin both are basic dye, and because this bacterial cell wall, it is negative charge. So initially, both these gram positive and gram negative bacteria both will take the, even if you stain it with crystal violet and or if you stain it with saffronin, both the type of bacteria will take up the stain. But yeah, there will be issues with the successive steps that will do. As you mentioned, that iodine forms a better complex with crystal violet rather than saffronin. And okay, so now let's move to nucleus. In bacteria, in this prokaryotic structure, what else was were taught? Mainly, the bacterial cell wall was the main thing that was taught in greater detail. Other things, flagella and these things you can go through any standard book and learn about them. Um, Now we will move to the eukaryotic cell. So the eukaryotic cell, one of the major organelle is the nucleus, right? So nucleus is the cell's control center and storage of the cell's genetic information. So if this is the structure of the nucleus. This figure I've taken from the book Cooper, and you see this nucleus is bounded by a nuclear envelope, and the nuclear envelope is composed of an inner membrane and outer membrane, and there are pores between these nuclear membrane, which are called the nuclear pores. Okay, and these nuclear pores have have a type of protein complex. Okay, they have type of protein complex which are called the nuclear pore complex. These are called the nuclear pore complex, or they are also called the nucleoporins. Nuclear pore complex. And what is the function of these nuclear pore complex? The nuclear pore complex, it regulates substances from or it regulates the entry and exit of substances to and fro from the nucleus. So, for example, you have a protein which is present in the cytoplasm, or you have a transcription factor which is getting synthesized by the ribosomes which are present in the cytoplasm, and you want this transcription factor to act on the DNA which is present in the nucleus. So in that case, the movement of or the entry of these protein structure of these protein molecules into the nucleus will be regulated by the nuclear pore complexes. Similarly, if you the RNA, the mRNA which is synthesized in the nucleus, you want this mRNA to move into the cytosol for translation. So the movement of mRNA or any other macromolecules outside the nucleus and into the nucleus are regulated by this nuclear pore complex. And then there is a dense structure which is present in the center of the nucleus, which is the nucleolus, and the DNA. The DNA is present inside the nucleus, and this DNA is not freely it is not free. It is not naked, right? So the DNA remains wrapped around histone proteins and forms nucleosome, and then this nucleosomes are form an array of nucleosome forming the structure which is known as chromatin. So chromatin is basically DNA which are wrapped around the histone octamer complexes. So and the outer membrane is continuous with the rough endoplasmic reticulum. Okay, yeah. So are this point clear? And just if you see inner to the on the inner side of the nuclear membrane, inner nuclear membrane, there are structures which are called the nuclear lamines. So anyone say the function of nuclear lamine or nuclear lamina? So, inner to the just inside the cell membrane, you have structures like actin, right? So that provides the shape and structure to the cell. So, this structure to the nucleus is provided by this nuclear lamina. Okay, so nuclear lamina present underlying the inner nuclear membrane, and it provides structural framework to the nucleus. All these points you have to remember about this nucleus. So these points are clear, right? They are also very basic. So you remember the function of these nuclear pore complexes. They are present. They allow the regulated transport of small molecules, ions, macromolecules, and the nuclear envelope separates the nucleus from the cytoplasm. Okay, and what are this chromatin? So if you stain the nucleus or inside, then you will find that few regions of the nucleus are stained very darkly, and few are stained light. So of the chromatin. So basically, if you isolate the chromatin from the nucleus, and if you stain it, or what you will find that there are some darkly stained structure, and then there are some lightly stained structures. So the structures which stain very dark are called the heterochromatin. They are stained very dark. Okay, and why are they stained dark? uh, Light stain. So in these region what happens, the chromatin is very is packed, the chromatin is very yeah, is condensed, it is very tightly packed. And because the nucleosomes, if you see the structure of that, they come in very close contact with each other, and that is why they stain dark, and these regions are called the heterochromatin. So these are the regions where the gene is transcriptionally active, which means that in these regions the transcription process will not take place because the DNA is very tightly packed. Okay, and the regions which are lightly stained are called the euchromatin, and they are transcriptionally active. So if you look at the structure of the nucleus, then the periphery of the nucleus mainly stains very dark because of the heterochromatin structure, and the inner regions of the nucleus, they stain light, and here the DNA is loosely packed, and that is why this region, the inner regions of the nucleus are called euchromatin, and the periphery, these regions have very tightly packed DNA, and they are called the heterochromatin. Okay, heterochromatin is highly condensed and not transcribed. And for example, you can see the centromeres and the telomeres. So if you look at the structure of chromosome, then you will see that mainly the central region of the chromosome, which is called the centromere, and the terminal ends of the chromosome, which are called the telomeres, they have repetitive sequences, and these regions stain very darkly, and these are the regions where heterochromatin is present. Okay, okay, now to answer this question, the significance of euchromatin and heterochromatin in terms of gene expression. So this is very easy concept to remember. So the structure which is very tightly packed, that will be inactive. That in that regions the gene will not be expressed, and the regions of the DNA which are loosely packed or which are free, the in those regions transcription will take place. Or yeah, so tell me the answer. Yeah, option B. Okay, so everyone agrees with option B. So yes, the euchromatin is less condensed and active, while heterochromatin is condensed and inactive. Okay, so answer is option B. Now, what is the function of the inner membrane of nuclear envelope? Function of the inner membrane of nuclear envelope is structural support, mRNA synthesis, DNA replication, initiation, regulation of nuclear pores. So, structural support is provided by which structure? They are provided by nuclear lamina, right? That is not the function of nuclear envelope. Now, mRNA synthesis is done by what? You will learn about this mRNA synthesis, transcription, translation. All these you will learn in subsequent lectures, right? So who are mainly involved in mRNA synthesis? Right, the RNA polymerases, right? The RNA polymerases will come, they will transcribe those regions. The RNA polymerases are involved in mRNA synthesis. The nucleus does not have any role. DNA replication initiation, I think you have basic class 11, 12 biology, you all know there are ORI sites, right? Origin of replication. The ORI sites which are present in the DNA. So there are particular DNA sequences at which the DNA replication will start. So these are called the ORI sites. So that also has no that has nothing to do with nuclear envelope, right? So you can eliminate the options and then you can select the correct answer if you are having any confusion. Okay. So structural support is provided by nuclear lamina. So option A is wrong. mRNA synthesis is done by RNA polymerase. Option B is also wrong. DNA replication initiation starts at ORI sites. You will learn about these things, okay, in more detail. So you don't need to worry now. And option D is regulation of nuclear pores. So that is the only option, and so option D is correct. Okay, because the inner membrane will control which molecules can inner membrane will remain attached to these nuclear pore complexes, right? So they will regulate what type of molecules can enter and exit. So you can read about the structures of nuclear pore complex. They are given in very detail in any standard cell biology book. You can read about these nuclear pore complex structures. They are multi-protein complex protein complexes. Option D is the correct answer. Okay. Now tell me the function of. Oh, so this is again a repetition. Just skip this one. I don't know how. Yeah. So now answer this question. Question number 7. Which of the following about nucleus is true? So you have to this. These are the options. Okay. A, B, C, D. So these are some statements which are given. 1, 2, 3, 4, 5, and these are the options. A. Okay. So let's read the statement one by one. The euchromatin is uncoiled, light, and genetically active. So is this statement true or false? First statement. First statement is true. Yeah, it is true. So euchromatin, it is uncoiled, it is light, it is genetically active. So now coming to statement two, heterochromatin is dark, firmly packed, and genetically inactive. This statement, this is also true. Yeah, this is also true. The heterochromatin is firmly packed, light. So this is wrong. Heterochromatin is not light; it is dark, and it is not genetically active. So this is false. This is also wrong. This is false. Okay. Option number the statement number four. Statement four. Euchromatin is uncoiled, dark. It is not dark. It is light, and it is genetically inactive. So it is light. So that is why option four is false. And option number five. Nucleoporins. So nuclear pore complexes are all only called nucleoporins. So nucleoporins is the other name of nuclear pore complexes. Okay. Nucleoporins helps in transporting proteins and an mRNA. Okay, so yeah, just now I was telling the function of nuclear pore complexes. They regulate which molecules can enter or exit the nucleus. So that is why this option is correct. Yeah, so option number A, 1, 2, and 5. Yeah. Okay. Oh, I forgot to add an animation here. So what? Anyway, the component of the nuclear envelope is directly involved in recognition and transport of proteins into the nucleus. Into the nucleus. So what is that structure present in nucleus which which can regulate or which recognizes and transport proteins into the nucleus? So these are these structures. You see this blue color. These are the nuclear pore complexes. Nuclear pores. Okay. So the nuclear pores are the components which are involved in. Okay, now coming to mitochondria. Okay, so everybody has read about the structure of mitochondria, right? So, mitochondria, they are double membrane structure. They also have an outer membrane and an inner membrane, right? They have an outer membrane, they have an inner membrane, and between the two membrane there is a space which is called the intermembrane space. And then this inner membrane is folded folded into finger-like projections. These finger-like projections, what are these called? These are called the cristae. These are called the cristae. And then inner to this inner membrane, you have the mitochondrial matrix. So matrix is the mitochondrial matrix is the fluid which is present in the inside of the mitochondria. Okay, so this matrix have different enzymes. They have different enzymes which are involved in these pyruvate to acetyl CoA fatty acid to all these metabolic reactions which are taking place inside in the matrix of the mitochondria. So different cycles take place, right? You must have read in biochemistry TCA cycle. Okay, so this TCA cycle, then fatty acid metabolism, different cycles, all these enzymes for converting one metabolite, and all these enzymes are present in the mitochondrial matrix. And the inner membrane, is very important because the electron carriers, electron transport proteins, all the electron transport proteins are present in the inner membrane. So you must have read about the complex 1, complex 2, complex 1, complex 3, 4, 5. So all the proteins which are involved in this electron transport system, and the process of oxidative phosphorylation. So they are present in the inner mitochondrial membrane. Okay, so yeah, different citric acid cycle this is taking place. And so you just remember these points. A few points you have to keep in mind, that this inner mitochondrial membrane have different proteins of the electron transport chain. And also this ATP synthesis is also present in the inner mitochondrial membrane, and the matrix have different enzymes which are involved in the TCR in the citric acid cycle. Okay, so it is a double membrane, inner and outer membrane, and the inner membrane is mostly impermeable to ions and small molecules. So remember this point: the inner membrane is impermeable. Why is it impermeable? Because this inner membrane is involved in a very important function. Okay, it is involved in a very important function in maintaining a proton gradient. It is involved in maintaining a proton gradient across the inner membrane. So that is why the inner membrane is impermeable to ions and small molecules. So if you have to maintain this proton gradients, you know, just now, we will learn about this electron transport chain in the upcoming slides. So then you will understand this point in more detail. So the outer membrane is permeable to small molecules, but the inner membrane is impermeable to small molecules. And the outer membrane, why is it permeable to small molecules? Because it contains channel proteins. One type of channel protein is present in the outer membrane, which are called the porins. And these porine proteins allow the free diffusion of molecules which are less than 100 Dalton. 100 Dalton to 5 5 kilodalton. So I think in the slides it was mentioned 5,000 Dalton. The inner membrane forms cristae which extend into the interior, and because of these finger-like projections, it gives a greater surface area, more surface area, so that more proteins can be accommodated here, and the efficiency of this electron transport system or oxidative phosphorylation can take place. Okay, and the matrix of mitochondria contains enzymes for TCA cycle and oxidative breakdown of fatty acids. The oxidative phosphorylation takes place in the inner mitochondrial membrane. So you all know about this. Mitochondria, and another important point is that mitochondria is inherited from mother, right? Uh, so 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 I think you all know about about this point, right? So during fertilization, what happens? Only the nucleus of the sperm enters the ovum, right? The all other structures which are present in the sperm, that gets degraded. So the mitochondria that is present in our cell, those are specifically inherited from our mother, because the ovum contains the mitochondria, while the mitochondria gets degraded from the the mitochondria cannot enter the ovum. And they are endosymbiotic. So you must have learned about this endosymbiotic origin, or and they are considered to have evolved from bacteria, because if you look at the structure of mitochondria and bacteria, they are very similar structures. They also have this have two membranes, and all these they have inside the matrix, they have their own genome, mitochondrial genome, so they can replicate on their own. So all these points. make this mitochondria. They have endosymbiotic organelles. Okay. So anyone has any doubt? Oh, I forgot to add the animation. So how do the small molecules pass through the outer membrane of mitochondria? So the answer is porins. So porins are channel proteins. They are channel proteins which are present specifically in the outer membrane of mitochondria. And these porin proteins allows the small molecules move move into the intermembrane space. Okay, into the intermembrane space, but the small molecules are not allowed to enter the inner membrane. Okay, the evolution of bacteria is characteristic to the evolution of which eukaryotic organelle? So yeah, so often it is considered that this mitochondria used to have an independent existence, like a bacteria, but then this mitochondria got engulfed by an eukaryotic organism, and then these mitochondria and the eukaryotic cell, they by the process of symbiosis, so the eukaryotic cell obtained a few benefits from the mitochondria. It provided it with energy, while the eukaryotic cell helped it in uh, transportation and shelter. So that is why that is the endosymbiotic theory. So that is why the bacteria evolution of bacteria is characteristic to the evolution of mitochondria. Okay. The world's smallest natural motor is present in which organelle? Yeah, so that is also mitochondria, because mitochondria provides the energy. It can it is involved in yeah, ATP synthase. So that is why it is the smallest natural motor. Okay. Now chemiosmotic hypothesis does not involve. So uh, okay, so let's first learn about chemiosmotic hypothesis. Okay. Mm. So first let's learn about these the electron transport chain. So what is the electron transport chain and chemiosmotic theory? So anyone? So this things is actually related to glycolysis and the TCA cycle, or the citric acid cycle. So in the glycolysis and so I don't know if you remember uh, these metabolic pathways, right? So in the glycolysis and in the citric acid cycle, these are produced this reducing substances NADH and FADH2, right? So this NADH and FADH2, when these molecules are oxidized, the proton which is released, the proton which is released is pumped or it is pumped into these intermembrane space, and when the and as a result, what happens is because of the presence of this electron transport proteins, there are several electron transport proteins which are. So yeah, look at this figure. I'll start with this figure. So you see the inner mitochondria. So this is the inner mitochondrial membrane. Okay. So these things I think I've also gone through in class 12 also. So this is the inner mitochondrial membrane. This is the outer mitochondrial membrane, and this is the intermembrane space. Intermembrane space. So as I was just mentioning, that the inner mitochondrial membrane has the proteins which are involved in the electron transport chain. So there are different protein complexes. The complex 1, complex 3, complex 4, and uh, and another is the complex 5. Okay, so what happens is when this the NADH and FADH2, these are produced in the glycolysis. So if you remember the glycolysis cycle, then you will remember that there are during glycolysis NADH, there are two molecules of NADH that are produced. Okay, two molecules of NADH that are produced in glycolysis. Then in the TCA cycle also there are around six molecules of NADH are produced in the TCA cycle. Okay, and between these there is one acetyl CoA pathway. So in that also there are two NADH molecules produced. And in the TCA cycle, along with this NADH, there are two molecules of FADH2 also are produced. So these molecules are oxidized. They get oxidized, and once they are oxidized, the NADH is oxidized into NAD plus and H plus, and these protons that are produced, these protons are actively pumped. They are actively pumped from the matrix of the mitochondria to the intermembrane space. Okay. And the electron that are produced, they are transported from one complex to the other complex. So there is this coenzyme Q. This coenzyme Q transports this electron from complex 1 to complex 3, and all these complexes, they have increasing reduction potential. So the complex 3 has more higher redox potential, or it has a greater tendency to accept electrons. So once the electrons pass from one complex to the other complex, the protons are also pumped from the matrix to the intermembrane space. They're actively pumped into the intermembrane space, and the electrons, they flow from complex 1 to complex 3 and then to complex 4. So, uh, so one pair of electron transport four protons into the intermembrane space. So, as a result, you can see that there are large number of proton molecules which gets accumulated in the intermembrane space. So, there are many proton molecules which get accumulated in the intermembrane space, and there is a shortage of protons in the matrix. There are The matrix has lower proton, the concentration of protons in the matrix decreases, while there is a high concentration of the protons in the intermembrane space. And now when these so when this gradient is broken, so when this gradient, how is this gradient broken? So there is this complex 5, the ATP synthase. So this ATP synthase transports this proton, or it breaks the proton gradient by transporting this proton from the intermembrane space into the matrix. And when this electrochemical gradient is broken, the ATP molecules are generated. From ADP you get ATP molecules. Okay, so this is how the electron transport system functions in the mitochondria. Okay. So is this point clear? So yeah, we will look at this. We will read each point one by one so that you understand in more detail. So the NADH and FADH2 molecules produced during glycolysis and TCA cycle are used for the synthesis of an additional 34 ATP molecules by the process of oxidative phosphorylation. So you know there are two types of phosphorylation: substrate level phosphorylation and oxidative phosphorylation. So by substrate level phosphorylation, you get how many ATP? You get 2 ATP. Net ATP that you generate, that is generated during glycolysis, is 2 ATP. Okay, so when one substrate is converted into another substrate, the phosphate, for example, this phosphoenol pyruvate, when it is broken into pyruvate, then you get one molecule of ATP. Okay, so that is substrate level phosphorylation. But these molecules that are produced, NADH and FADH2, when these get oxidized, and you get ATP molecules. So from these molecules, when you get ATP molecules, this is called oxidative phosphorylation. Okay, so the oxidation of NADH and FADH2 releases electrons which are carried by a series of electron carriers present in the inner mitochondrial membrane, constituting the electron transport chain. Okay, so yeah, so when these molecules NADH and FADH2, when these are oxidized, then what happens? The electron, because oxidation will lead to what? It will lead to loss of electron, or electrons will be generated, and these electrons are transported from one electron carrier to the another electron carrier, and to the successive electron carriers. And when these electrons are transported from one electron carrier to another electron carrier, the large amount of free energy is released, and this large amount of free energy which is released is used for the transport of protons from the mitochondrial matrix to the intermembrane space. Okay, so as I was mentioning that the redox potential or the complex 3 has a greater has more tendency to accept electrons than the complex 1. So as a result, what happens when this complex 3 accept electron, it releases large amount of free energy, and this free energy that is released is used for transporting the proton from the matrix into the intermembrane space. And as a result, what happens? An electrochemical gradient. So you see a concentration gradient is generated between the intermembrane space and the matrix. So you see the protons, they get accumulated in the intermembrane space, while there is a few number of proton molecules in the matrix. And also because of the as the protons have charge, there is also an electric potential. An electric potential is also generated between the intermembrane space and the matrix. And so what happens when the flow of protons down the electrochemical gradient through the complex 5 or ATP synthesis is coupled to ATP synthesis. Okay, is coupled to ATP synthesis. So now answer these questions. So now tell me, what is chemiosmotic hypothesis? So just now, what I was that I discussed just now, that is the chemiosmotic hypothesis, which suggests that the flow of hmm, yeah, okay. Any more answer? Option D, yeah. Any other answer? So chemiosmotic hypothesis is what? So whenever this electrochemical gradient is broken, it is coupled to the synthesis of ATP. Okay, so that is what is called the chemiosmotic hypothesis. So only proton transport is, so let's read out the statements one by one. So only proton transport is strictly regulated. Other positively charged ions can diffuse freely across the mitochondrial membrane. So this option correct or wrong? So you see, only protons transport is strictly regulated. Other positively charged ions can diffuse freely. So the very first point that I mentioned in the slide, so that is that the inner membrane very tightly regulates the movement of small molecules and ions, because it has to maintain this proton gradient. That is why the inner membrane does not allow any type of positively charged ions to enter into the matrix, because if for example, if it allows potassium ions or sodium ions, then this electrochemical gradient will be disturbed, and then the ATP will not be generated. So that is why this the second part of this statement is actually wrong. Okay, the ATPase activity is, so you read this question very carefully. Okay, chemiosmotic hypothesis does not does not involve, it is not what it involves. The question is, it does not involve. So option A is false. Okay, ATPase activity is reversible. ATPase activity is reversible. This statement is also true. You can convert ATP to ADP, and if you add another phosphate group, you can also form ATP. So the ATPase activity is reversible. This statement is true. Proton flow into the mitochondria depends on the presence of ADP and PI. So this is also true, because once the proton is getting transported from the intermembrane space to the matrix, if there are no ADP and PI molecules, if there is a lack of these molecules, then the ATP will not be generated, right? So it will have to depend on the presence of ADP and PI, inorganic phosphate. And the fourth option, electron transport by the respiratory chain pumps proton out of the mitochondria. So this statement is correct. So that is what we were learning in the electron transport chain, that the transport of, so the electrons are flowing from one electron carrier to the other, and when these electrons are transported from one electron carrier to other, what happens? There is free energy released, and this free energy is involved in pumping the protons out of the mitochondria. The protons were getting pumped from the matrix side to the electron transport carriers. What were they doing? They were transporting the protons from the matrix to the intermembrane space. Okay. So option number D, these are all true statements. So the question is, it does not involve. So option number A is wrong. Okay. I hope you understood the solution to this question. Okay. You read the question very carefully. Okay. So read the first statement. Only proton transport is strictly regulated. Other positively charged ions can diffuse freely across the mitochondrial membrane. So you tell me whether this statement is correct or incorrect. So just now what we were seeing. So you see here. So the transport of protons are strictly regulated. That is true. But but if the transport of any other ions, for example, there is K plus ion or Na plus ion, those type of ions, transport of these type of ions has to be regulated very strictly, right? If these ions are if the movement of these ions are not regulated, then this electric potential will be disturbed, right? This electric potential will be disturbed, and then the movement of these protons will cannot be coupled to ATP synthesis. So that is why statement number A is false. So here you see other positively charged ions can diffuse freely across the mitochondrial membrane. So this statement is false. It is uh, other the positively if any type of positively charged ions can diffuse freely, then the potential difference of the electrochemical gradient that will be disrupted, right? Okay. So you see here, this point I was emphasizing on this point for this reason, that it is inner membrane is mostly impermeable to ions and small molecules. The outer membrane is permeable to ions and small molecules, but the inner membrane is almost impermeable. And why is it so? Because it is essential for maintaining the proton gradient. Okay. Now is it clear? Now, option number B, option B, option C, and option D, they are all correct, right? ATPase activity is reversible. This is correct statement. Proton flow into the mitochondria depends on the presence of ADP and PI. This is correct. So, did you get this question? Okay. This is again repetition. Yeah, now tell me the answer to this question. The main driving force for the rotation of ATP synthesis rotor during ATP synthesis is, so what? Proton gradient, yeah, correct. So what happens when this proton gets transported from the intermembrane space into the matrix? This F naught F1 particle, you must have learnt about this, right? This is the F naught and this is the F1 particle. This F naught can rotate. So when this proton moves from this intermembrane space to this matrix, this F naught particle, this subunits, they can rotate, and this allows for the. So in lecture one, if you had gone through lecture one, Vishal sir had shown a very nice animation of how this ATP synthase is involved in transporting these protons and how the ATP is generated. Okay, so yeah, the answer is correct. Answer is proton gradient. Yeah. Now tell me the answer of this question. ATP synthesis generates ATP during oxidative phosphorylation by. Now read the statements very carefully. Okay. Directly phosphorylates ATP. So you take a few minutes, 2 minutes, 1 or 2 minutes, and then all of you tell me the answer to this question. We can also generate a poll. I will, from next class, what I will do, I will generate a poll, and then we can discuss the answer. So that will be more fun to. Okay, someone is saying A, B. ATP synthesis generates ATP during oxidative phosphorylation by. So just now I was discussing about substrate level phosphorylation and oxidative phosphorylation, right? So you have to understand the difference between substrate level phosphorylation and oxidative phosphorylation. So what is substrate level phosphorylation? So for example, there is phosphoenol pyruvate. So if you remember the glycolysis pathway, Then in the last step of the glycolysis pathway, phosphoenol pyruvate gets converted to pyruvate, right? Phosphoenol pyruvate gets converted into pyruvate, and so this phospho group, what happens when this phosphoenol pyruvate gets converted into pyruvate, this phosphate groups gets transferred into the ADP molecule. So this phosphoenol pyruvate, what it does, it transfers this phosphate group from the phosphoenol pyruvate to the ADP, and the ATP is generated. Okay, so that is called substrate level phosphorylation. So this is one reaction where for substrate level phosphorylation takes place, and there is another reaction. So 1,3 bisphosphoglycerate, when it gets converted into phosphoglycerate. So these are all reversible reaction. So when this 1,3 bisphospho, this is another step in the glycolysis pathway where 1,3-bisphosphoglycerate gets converted into phosphoglycerate. Here also what happens, so there are two phosphate molecules, right? So when one of these phosphate molecules, so you see here there are two molecules in bisphosphoglycerate and phosphoglycerate. There is only one phosphate molecule. So what it does, it transfers this phosphate group to ADP, and hence ADP gets converted into ATP. So these are substrate level phosphorylation. But ATP synthase does not work. ATP synthase, how does it generate ATP? So it is ATP synthase couples the when couples the proton gradient, when this proton gradient or along the proton concentration gradient, when proton flows, it couples this energy that is released for the synthesis of ATP, right? So that is oxidative phosphorylation. So you see here option number A directly phosphorylates ADP using energy from electron transfer. So this option is not correct. Directly phosphorylates ADP using energy from electron transfer. So yeah, using the energy from electron transfer, what was happening? The proton molecules were getting transferred from the matrix to the intermembrane space, right? And now ATP synthase, what is ATP synthase doing? ATP synthase is translocating when the protons are moving or getting translocated down their concentration gradient, that was getting coupled to the ATP synthesis, right? So the energy that was released from electron transfer, that was used for transporting the protons from the matrix to the intermembrane space, right? But ATP synthase, what it was doing, it is using this concentration gradient of protons for ATP synthesis. So option number B is correct, right? Did you get this point? So I will discuss again. So here you see when these electrons are getting translocated from one complex to the other complex, the free energy that is released, that is used for transporting the proton. So this free energy is for transporting the proton from the matrix to the intermembrane space, right? Here also complex 3 is also doing the same thing. It is using this free energy for transporting proton from matrix to intermembrane space. Complex 4 is also doing the same thing. But what is ATP synthase doing? You see ATP synthase is using this when this proton is translocating along the concentration gradient, then it is using this concentration gradient for generating ATP. So that is why the answer to this question is option B. So the translocation of protons down their concentration gradient is used for the generation of ATP. Okay. So is this point clear? So option number B. Okay. Anyone has any doubt in this question? Hmm. Option number A, that is what I'm saying. Why is option number A incorrect? It is because directly phosphorylates ADP using energy from electron transfer. So it is not using the energy from electron transfer for phosphorylating ADP. It is using the energy that is released when the proton is transported, when the proton is is getting transported from the, or it is breaking when the proton gradient is getting broken. So there is a large amount of free energy that is released, and that free energy is used for generation of ATP. So this second part of this statement, no, this energy from electron transfer, the energy the energy from electron transfer is used for transporting these protons, protons from the matrix to the intermembrane space. This energy is not used for generation of ATP. ATP is not generated by this energy. Energy ATP is generated due to the when you are understanding, right? When this proton gradient is broken, the free energy that is released, that is used for ATP generation. So this statement using energy from electron transfer, this statement is wrong. Okay, so that is why I had mentioned this point. You see here. So this point, third, fourth point, if you read this point carefully, the large amount of free energy released is used for transfer of protons from the mitochondrial matrix to the intermembrane space. Okay, it is not used for generation of ATP. For the generation of ATP, the protons has to flow down the electrochemical gradient. So this electric potential that is generated, when these protons will flow along this electric potential, or this electric potential and the concentration gradient that is generated, then only ATP will be generated. So that is the basis of chemiosmotic or chemiosmotic theory. Okay. The final electron acceptor in the electron transport chain. So this is another very important question, which you get everywhere, right? Option C, yeah. Many of you will know this answer because this question is asked frequently. So you see here, so the ultimately, if you see this diagram carefully, you see the electron is getting accepted by this proton. The oxygen is accepting the electron, and water is being formed. Okay, so that is why the final acceptor of electron in the electron transporter chain is oxygen. Okay, final electron acceptor is oxygen. Now tell me this point answer. During ATP generation in the mitochondria by ATP synthase, proton moves from where does the so you read the question carefully. ATP generation by ATP synthase. So how for ATP generation by ATP synthase, proton will move from which side? Option A. Anyone? Any other answer? Yes, the answer is option A. From the intermembrane space to the matrix. Okay. Because the protons were pumped into the intermembrane space and the electrochemical gradient was generated, and when this electrochemical gradient is broken, the free energy that is released is used for ATP synthesis. So the answer is from intermembrane space to the matrix. That is the function of ATP synthesis. It pumps proton from the intermembrane space to the matrix. Okay, now okay, this is another again repeat. Now let's learn about the structure of chloroplast in more detail. So so there were many things that were discussed in week one. So if you have doubt in any particular section, then I can take up those section. But I'm what I'm doing is I'm covering the important parts of the week one. So the parts which are more complicated and which requires more understanding. So I'm covering those parts only. The other parts that were covered, like media preparation and all those, there is nothing much to understand in those parts. But still, if you have any doubt, then I can discuss those also. Okay. So yeah, so chloroplast is another organelle which is very similar to the mitochondria in the sense that it has an inner membrane and an outer membrane, and between the inner membrane and outer membrane there is a space, intermembrane space. And another striking feature of this chloroplast is that it has another membrane system. Another membrane system is present inside the chloroplast, which is formed by this thylakoids. So you see this disc-like structures which are present inside the chloroplast. These are called the thylakoid. These are called the thylakoid, And when these thylakoid or these sac-like structures, what happens is these thylakoids get arranged one on top of the other, and they form a stack of thylakoids. A stack of thylakoid is called what? It is called a grana. It is called granum. Singular is granum and plural is grana. So a stack of thylakoid is called a grana, and you see the one grana remains attached to another grana via this, what is this called? So these are the these are also connected to one another. Okay, so these structures connect one grana to another grana, and the matrix, or the matrix which is present inside the chloroplast is called the stroma. Okay, the matrix present inside the chloroplast is stroma. So chloroplast are, all of you know, they are organelles responsible for photosynthesis. They have an inner membrane, outer membrane. So this is inner membrane, outer membrane. There are thylakoids, and the matrix which is present is called stroma. There are three chambers which are present. So if you see, there are three chambers that are getting found. One is this intermembrane space. Then there is this stroma, and another chamber which is being found is the thylakoid lumen, right? The other thylakoid lumen. And the thylakoid membrane, so like we were discussing in mitochondria, the inner mitochondrial membrane, where the inner mitochondrial membrane had different proteins which are involved in electron transport chain, right? So similarly here the membranes which are having these different photosynthetic pigments, they are the thylakoid membranes. So you remember, the thylakoid membrane contain chlorophyll pigment, components of electron transport chain, and light absorbing pigments. So if you have read biology in class 12, then you must have read about this PS2, PS1, and the cyclic transport of electrons, non-cyclic transport, all these things you have studied, right? So all the different pigments, the PS1, PS2, and pheophytin, and all these different molecules, these are present on the thylakoid membrane. And the stroma contains enzymes involved in carbohydrate synthesis. So you must have read about the light reaction and dark reaction. So the light reaction takes place in this thylakoid membrane, right? Because this thylakoid membrane have different proteins which are have different photosynthetic pigments. Okay, while the dark reaction. So what is the dark reaction? When these NADPH and ATP are used for synthesis of glucose, right? Okay, the ATP and NADPH is used for generation of glucose. So that is the dark reaction. So all the different enzymes, Rubisco, and the different enzymes which I have studied, all those different enzymes are present in the stroma of the chloroplast. Okay, so let's learn about this process very quickly. We will go through this. Similar to what we were studying in mitochondria, here also the same thing is taking place, because chloroplast will also help in the generation of ATP. Okay, so in the this is the light reaction. In the light reaction, so the photon gets first they are get it gets absorbed by the PS2 photopigment, and then this PS2, the energy that is absorbed is used for the photolysis of water. So photolysis of water takes place in PS2. The water gets hydrolyzed into protons and oxygen, and the electron that is released, it gets transferred from photosystem 2 to cytochrome BF6 complex via this plastoquinone. Okay, so this pathway you have studied, I think. So then this electron is transferred from cytochrome BF complex to the PS1 via the plastocyanine, and ultimately the electron is used for generation of this NADP. NADP reductase. NADP reductase accepts the electron and helps in the reduction of NADP to NADPH. Okay. So all these different pigments that you are seeing, these PS2, PS1, cytochrome BF, they are present where? They are present on the thylakoid lumen. They are present in the thylakoid lumen. And here what is happening, so when these electrons are getting transferred from one complex to the other complex, the protons are pumped from the stroma. They are pumped from the stroma into the thylakoid lumen. Okay, so there the proton was getting transferred from the matrix to the intermembrane space. Here the protons are getting transferred from the stroma to the thylakoid lumen. And then when this proton, so similar thing here also, the chemi-osmotic theory is only happening. Okay, so once this proton gradient is broken, or when the proton flows along their electrochemical gradient, the ATP is generated. So ATP gets generated in the stroma. So you remember that NADPH, ATP, all of these are getting synthesized in the stroma. Okay, and the ATP synthesis, what it is doing, it is transferring, translocating proton from the thylakoid lumen into the into the stroma of the thylakoid. Okay, so yeah, let's read these points. So there are four protein complexes in the thylakoid membrane, and these function in electron transport and synthesis of ATP and NADPH. So all these different protein complexes, what are they involved in? They are ultimately, their function is what? Their function is generation of NADPH and ATP, so that this NADPH and ATP can be utilized in the dark reaction for the synthesis of carbohydrate, which is the food of the plant. Okay, for the synthesis of glucose. So what happens at photosystem 2, the energy derived from photon absorption is used to split a water molecule within a thylakoid lumen. So the splitting of water molecule takes place in the thylakoid lumen. Okay, so remember these points, these are important points. Statements can, direct statements can come from these parts, and you will be asked to tell which is the correct option, or which of these statements are correct. So you understand these points very clearly. So at photosystem 2, the energy derived from photon absorption is used to split water molecule. Within the splitting of water molecule takes place in the thylakoid membrane and the thylakoid lumen, and then the electrons are carried by plastoquinone. They are carried from plastoquinone to cytochrome BF complex, and the protons are pumped into the thylakoid lumen. So you see the protons are getting pumped from this stroma to the thylakoid lumen. Okay, now electrons are then transferred to photosystem 1. Okay, so you see the electrons are first, they are getting transferred from photosystem 2 to cytochrome BF by this plastoquinone, and then the electrons are further transferred from cytochrome BF to PS1 via the plastocyanine. Okay, at photosystem 1, what is happening? The energy derived from light. So you remember that the photosystem 1 is not involved in pumping proton. Okay, you see the photosystem is not pumping any proton. Okay, so what it is photosystem 1 doing? Photosystem 1, the energy, it is again absorbing energy. So there are two different wavelengths of light which are absorbed by photosystem 2 and photosystem 1. The photosystem 1 absorbs 700 nanometer of light, and this absorbs around 680 nanometer light, right? So these points you must have read in class 12 also. So electrons are transferred to photosystem 1. At photosystem 1, the energy derived from light absorption again generates high electrons, which are transferred to ferrotoxin. Ferrotoxin, so FD you see here, this molecule uptakes this electron, and it transports it to the NADP reductase. So the NADP reductase is the enzyme which reduces the NADP plus to NADPH in the stroma, and ATP synthase then uses the energy stored in the proton gradient to convert ADP to ATP. So first it generates this proton gradient is generated, and when this proton flows along this gradient, then the ATP will be generated. Okay, these points are clear, right? Am I audible? Okay, okay, okay. Okay, now answer these questions. The membrane within the chloroplast that contains photosynthetic pigments and is the site of light dependent reaction. This is a very simple question. Yeah, thylakoid membrane. Right now we were discussing these points only. So the thylakoid membrane is the is the site where all the different protein complexes PS1, PS2, cytochrome BF6, ATP synthase, all of these are present in the on the thylakoid membrane. And in case of mitochondria, it was present on the inner mitochondrial membrane. Okay. So these things you can study them in very detail, but for this course, so these are just the basics. So only the basics of cell biology, what are the different organelles. So I think in your BSc, MSc, you read these in much more detail. Okay, more detail than what is covered in the in this course. We will learn about molecular bio in more detail. So these are just to brush up your concepts. Now to answer this question, function of the stroma of chloroplast. Yeah. Option C, right? Because the stroma contains the different enzymes which are involved in the dark reaction. All the different enzyme Rubisco, and then there are different. This in entire. I think you remember the Calvin cycle. So RUBP, how is it getting converted into glucose? So all these different enzymes are present in the stroma. So the function of stroma is to carry out dark reaction of photosynthesis. So these are class 12 questions actually. Now, for, okay, so this is again repetition. I don't know why it's repeated. Now, organelles of vesicular transport. So this is another thing which was covered. So in eukaryotes, this vesicular transport is one of the is another aspect. So you know about this endomembrane system. The what does this endomembrane system does that is that the protein which gets synthesized. So you have the rough endoplasmic reticulum here, right? The proteins which are synthesized by ribosomes which are present on the rough endoplasmic reticulum, they move into this endoplasmic reticulum, and from endoplasmic reticulum, these proteins, here different types of protein modification takes place, disulfide bonds and other things take place here, and then with the help of these vesicles, with the help of these vesicles, these protein molecules, or it can be lipid molecules or any substances, they move from this endoplasmic reticulum to the Golgi complex, right? So you have this is Golgi, then trans medial Golgi, trans Golgi network. So all these concepts are there in any you can get them in any standard cell biology book. So from the Golgi complex, what is the function of Golgi complex? So Golgi complex is like the post office of the cell. Okay, what happens in a post office? There are different letters from different parts of the countries. We get different letters are received in the post office, and then what the post office does, it depending on where it has to get transported, it will transport it to, for example, there is a letter which has to be transported to suppose Mumbai, suppose Kolkata, and here in this way, also what is happening inside the cell. So there are different proteins. Some protein has to reach mitochondria, some proteins has to reach peroxisome, some has to reach outside the cell, it has to be secreted outside the cell. So that is the function of the Golgi complex. It is packing these proteins and transferring them to different parts of the cell through these vesicles. The vesicles bud out from these membrane, and they are secreted out either outside the cell, secretory pathway, or it reaches to other locations of the cell, to the mitochondria, to the lysosome, different parts of the cell. So the organelles of vesicular transport, they comprises of the endoplasmic reticulum, the Golgi complex, endosome, lysosome, vacuoles. Okay. So actually the lysosomes, what happens if any protein has to enter the cell, or the degradation, all these takes place in the lysosome. So protein synthesized in the endoplasmic reticulum get modified in the Golgi complex and transported to various destinations such as plasma membrane, lysosome, or large vacuole of the plant cell. Now, the endoplasmic reticulum. So the endoplasmic reticulum is a membranous, it covers a very large portion of the cell. The membrane of this endoplasmic reticulum covers the entire cell, and it is a network of membrane. It is a network of membrane enclosed tubules and sacs that extends from the extends from the nuclear membrane throughout the cytoplasm. So in the initial slides, I had shown you the structure of animal cells. So this endoplasmic reticulum, it remains scattered throughout, or it is spread throughout the structure of the cell, or throughout the structure of the throughout the cytoplasm, and this entire so these structures, these tubule-like structures, these are called the cisternae, and these are called the tubules. So these structures, these are called the tubules, cisternae, and then there are rough endoplasmic reticulum. So there are two types of reticulum. You all know about this: smooth endoplasmic, rough endoplasmic. Rough endoplasmic means the endoplasmic reticulum will be covered by ribosomes on its surface, and the smooth and because the endoplasmic reticulum remains surrounded by the ribosome, so it is involved in protein synthesis, right? It is involved in protein processing. And the smooth endoplasmic reticulum is involved in lipid synthesis. You all know about these points, right? The ER is the site of protein folding. Because for protein folding, one of the important bonds that is formed is the disulfide bond, SS bond. So this SS bond or the disulfide bond can only be formed in an oxidizing environment. So the oxidizing environment is actually provided by this endoplasmic reticulum. So the protein folding and disulfide bond formation, all these takes place in the endoplasmic reticulum. So remember the functions of these organelles. They are involved in assembly of multi-subunit proteins, disulfide bond formation, and one of the enzymes which is involved is protein disulfide isomerase. So that is one of the enzyme which is present in endoplasmic reticulum, and it helps in the formation of the disulfide bonds. N-link glycosylation. So if there is a protein and you need to add a sugar group, you need to add a sugar group to the protein molecule. These sugar molecules are added at specific residues, amino acid residue, which are the asparagine residue. You must have learnt about the O-link glycosylation and N-link glycosylation. So the N-link glycosylation takes place in the endoplasmic reticulum, and addition of glycolipid anchors to some plasma membrane proteins. So these are some of the functions. And Golgi complex, as I was mentioning, that it is also known as the post office of the cell. Okay, so what happens? So the vesicles, the proteins which are getting synthesized in the by the inside, or which is present in the endoplasmic reticulum. So the ribosomes, what it does, you must have learnt about how these proteins which are synthesized in the ribosomes, uh, they have different they have what are they called, protein tags, these are signals, localization signals. They have different localization signals, and based on which the Golgi complex will decide where this protein has to be sent, whether it has to be sent to mitochondria or whether it has to be secreted out of the cell. All the proteins which are getting synthesized by the ribosomes, it moves into the ER lumen, and then this ER membrane, it bulges and forms vesicles, and then these vesicles again fuse with the Golgi complex. So this is the cis Golgi. So cis Golgi is that Golgi which is present near to the endoplasmic reticulum, or which faces the endoplasmic reticulum, and which receives the vesicles which are budded out of the endoplasmic reticulum. And then this is from the cis Golgi, it moves to the median Golgi, and then to the trans Golgi. And what happens in the trans Golgi network? In the trans Golgi network, the different packaging happens in the trans Golgi network. Different based on so this trans Golgi network decides where this protein or where this vesicle has to be sent. Okay, so these things are in are this is not actually the focus of this course. So these you can learn in cell biology. They are in much detail. We will not go through so much detail. Just understand what are the different structures. Proteins received from the endoplasmic reticulum are further processed and sorted for transport to their eventual destinations. They can be transported to lysosomes, plasma membrane, or they can be secreted out of the cell. Okay, the Golgi is composed of flattened membrane enclosed sacs and associated vesicles. So you see here different vesicles are associated with the Golgi, and they have also these sacs. So these are also called the cisternae. These tubular sacs that you are seeing. These are called the cisternae, and the proteins from the ER proteins. This you try. you all know about this, the proteins from the endoplasmic reticulum, they will enter at the cis phase and they will exit from the trans phase, which is usually oriented towards the nucleus. So the cis phase remains oriented towards the nucleus and they are transported through the Golgi and exit from the trans phase, or is it also known as the exit phase. And the proteins are carried through the Golgi in the cis to trans direction with the Golgi cisternae. So this Golgi complex actually have a polarity. Okay, they move from the cis to the trans. So all the protein molecules, or they can be lipid molecules, it moves from the cis to the trans phase. And the modification of proteins, polysaccharide, lipids take place in the cis to trans Golgi network. So different types of modifications you must have learned about: mannose 6 phosphate modification, then O-link glycosylation also takes place in this Golgi complex. So different chemical moieties get added to the protein, salicylic acid, or there can be phosphoryl group which gets added to the protein molecule, or there can be other chemical groups, ubiquitin, or these types of modifications. All these different modifications and packaging will take place in the Golgi complex. Okay. Okay. Now answer this question. The following are the functions of the endoplasmic reticulum. So these are the options A, B, C, D. You need to read these statements very carefully. So what is statement number one? Statement number one is it is the site for protein synthesis for secreted and integral membrane proteins. 1, 3, 4, and 5. Okay, we will see if you are correct. So you read this statement. This is the most important statement actually. Which of the above are false? Okay. So if you don't read this, then even after knowing your answer will be wrong. Okay. So you have been asked to identify the false statement. Okay, false statement, not the correct statement. So these are actually tricky questions. You have to read the question very thoroughly. So you see, first question, first statement: site for protein synthesis for secreted and integral membrane protein. Anyone wants to try? So this first statement is it correct, or is it correct or false? This, yeah, this is true, because just now we saw that these RER, they are site for protein synthesis. Site of bulk membrane lipid biogenesis, is this true or false? This is also true, because the smooth endoplasmic reticulum, it is involved in lipid biogenesis. Then statement 3: proteins and phospholipids are transferred and biochemically modified in the region of the endoplasmic reticulum that is that is close to the Golgi apparatus. Yeah, so just now we were uh, no, this is actually true, right? Why will this be false? We just now learned, no, in Golgi apparatus what is happening? Different types of modifications were taking place, right? I told you about the phosphorylation, mannose phosphate, then there are ubiquitination. All these different chemical modifications, they are taking place in the Golgi apparatus. Glycoprotein, so if the carbohydrate moiety has to be added to the protein molecule, all these things will take place in the Golgi apparatus. So this statement is also true. Statement 4: major store of intracellular calcium. So this statement, you must have learned about this muscle contraction, relaxation in your class 12, right? The chapter. Um, this chapter was there. You learned about this actin, myosin. In this chapter in class 12, we have learned that there are smooth endoplasmic reticulum. These smooth endoplasmic reticulum are called sarcoplasmic reticulum, and these sarcoplasmic reticulum store calcium. So these are actually modified endoplasmic reticulum which store calcium in the muscle, and which is useful for muscle contraction. Okay, so this statement is also true. This statement is also true. Then option number 5, the ER is composed of, endoplasmic reticulum is composed of continuous membrane system that includes the nuclear envelope and the peripheral yard. This is also true. Just now we saw the diagram of nucleus, right? We saw that the outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. So that is what has been stated here. Now, option number six, question statement number six: the modifying, sorting, and packaging of protein for secretion. I think there is some error in this question. Uh, uh, this question is actually not complete. I will. I think this part is missing. Actually, the answer for this question is option number six, but I will make changes. I think this has something for secretion. This is incomplete statement. I will. Hmm, hmm. That is why this question is, yeah, for secretion. This is, yes, yes, yes. This is actually complete. Okay, this is done by Golgi bodies. Yeah. So statement number six is false. Okay. Is it clear to everyone? Yes, yes. Huh. Proteins and phospholipids are transferred and biochemically modified in the region of ER that is in close contact with the Golgi apparatus. So actually क्या है ना कि there are few modifications which take place in the endoplasmic reticulum and few modifications which take place in the Golgi. Okay, so in the endoplasmic reticulum, end-link glycosylation, endoplasmic reticulum end-link glycosylation take place. Then there are different signal peptide. So when a protein is synthesized, there are these N-terminal sequences. The N-terminal sequences of a protein which have certain specific sequences, specific amino acid sequences, which determine where the protein has to be transported, whether it has to be whether it will be a part of the membrane structure of the endoplasmic reticulum, whether it will be present in the lumen of the endoplasmic reticulum. So these signal peptides are actually cut. They are cut by the signal peptides which are present in the endoplasmic reticulum. So in the endoplasmic reticulum also certain modifications take place both in endoplasmic reticulum and Golgi complex. In both of these organelles protein modifications take place. Okay, so that is why this statement is actually correct. Proteins and phospholipids are transferred and biochemically modified in the region of endoplasmic reticulum. So one of these modification is N-link glycosylation. Then there is trimming of the signal peptides, or the signal peptides are cut in the endoplasmic reticulum. Then uh, then there are many types of modifications which takes place in the endoplasmic reticulum also. That is why this statement is true. Okay, is it clear now? Yeah, okay. So, peripheral, where it was peripheral, okay. The ER is composed of a continuous membrane system that includes the nuclear envelope and the peripheral ER. Yeah, so peripheral ER means the endoplasmic reticulum, which are in the periphery, or which are in the periphery region of the endoplasmic reticulum. So there will be few endoplasmic reticulum which will be very close to the nuclear envelope, right? And the endoplasmic reticulum extends up to the plasma membrane. So if you see this structure, I'll show you. Yeah, this structure you see, this is the structure of an animal cell. So these are called the peripheral endoplasmic reticulum because they are extending up to the periphery of the cell. Okay, so you see here the endoplasmic reticulum is it is present throughout the cytosol. It is spread throughout the cytoplasm. So so these are also endoplasmic reticulum. You see, so these endoplasmic reticulum which are present in the periphery of the cell. These are called the peripheral, and which are present near to the nucleus. They are the you can call them distal or near to the nuclear envelope. Is it clear? Okay. Okay. Now we will move forward. Um, Now there is not much. We have almost come to the end. Uh, these things we have discussed. Yeah. So to answer this question, the primary function of the Golgi apparatus in vesicular trafficking is, yeah, modifying and packaging proteins. So remember this that endoplasmic reticulum and Golgi apparatus both are involved in modification of proteins. Okay, modification of protein happens in both, but packaging of the proteins will specifically happen in the Golgi apparatus. Okay, Shandi. Now, plasma membrane. I have not discussed about plasma membrane, but I think you all know about the structure of plasma membrane, and this was covered in the lecture. Uh, so, uh, so I try to answer this question. Um, as the temperature increases, so which of the following? So you must have all. I think you know about the fluid mosaic model of plasma membrane and the fluidity of the plasma membrane, or this the concept that is related to fluidity of the plasma membrane. Is it known? So the plasma membrane have a hydrophilic head, hydrophobic tail. All these things have you read about? It was covered in the lectures because there is a time limit. I cannot go beyond 2 hours, so I have not discussed the plasma membrane. Uh, so let's discuss this question. Through this question, we will understand we will understand about the fluidity of plasma membrane. Okay. So as the temperature increases, so you all know that the plasma membrane is a uh, is it has a fluidic nature. Okay. And why does it have a fluidic nature? Because because if you see that the structure of plasma membrane, they are composed of lipid bilayer. They have a hydrophilic head and a hydrophobic tail, right? And these plasma membranes are very flexible in the sense that these lipid bilayers, these lipid molecules can interchange with each other. They can move from one leaflet of the membrane to the other leaflet of the membrane, or they can move laterally. They can undergo flip-flop motion. So uh, as the temperature increases, what will happen? It will increase. It increases fluidity and increases transport and of proteins and ions across membrane. Uh, uh, just give me a second. Uh, Hello. हाँ सर बोलिए, हाँ सर बोलिए। हाँ हाँ हाँ, चल ये मैं अभी और 5 मिनट है वो खत्म करके, बस वो लेट हो गया। वो ये लेट हो गया था ना स्टार्ट करने में। हाँ हाँ, ठीक है। हाँ हाँ, रिकॉर्डिंग तो हो रहा है। कोई बात नहीं। Okay, just a minute. I have two more slides. I will just quickly complete them. So yeah, so the correct answer for this question is actually what happens when the temperature increases, the fluidity of the plasma membrane increases, and it also leads to increase of transport of proteins and ions across membrane. And these lipid bilayers, if you see the structure of lipid bilayers, they have fatty acids. So these fatty acids, depending on what type of fatty acids are present, whether it is saturated fatty acid or unsaturated fatty acid, that will determine the fluidity of the plasma membrane. So saturated fatty acids, if the saturated fatty acids are more in number, then the then what will happen? If the saturated fatty acids are more in number, then the uh, fluidity of the plasma membrane will decrease. Okay, saturated fatty acids. So saturated fatty acids decrease membrane fluidity. Okay, decrease membrane fluidity. And unsaturated fatty acid, what it does is it increases the fluidity. So what are unsaturated fatty acids? So these things you will learn in biochemistry. So actually these unsaturated fatty acids have double bond. So there are double bonds between methyl groups, different methyl groups, and because of this double bonds, they can form a kink-like structure in the they can form a kink-like structure in the lipid in the structure of this lipid. Okay, so that is why the fluidity increases. So as the temperature will increase, what will happen? The temperature increases, it will increase the fluidity. That is true. And as the saturation of fatty acid increases, it decreases the fluidity of the membrane. So if the saturation of fatty acid increases, saturation means there are more number of single bonds. Okay, so as the saturation increases, it decreases the fluidity of the membrane. So this statement is also true. So 1 and 3 are correct. Okay, 1 and 3 are correct. Now, there is one more thing I wanted to cover. This is differential centrifugation. So, what is differential centrifugation is when you want to isolate the different components of a cell. So, different organelles. So, the cell has different organelles. So, now you want to specifically study the mitochondria, or you specifically want to understand the nucleus in a cell. So, all these things. If you want so if you want to isolate specific organelles, then you will go for differential centrifugation. So what is it? So basically first the cell is homogenized means it is disrupted, or the structure of cell is broken down. And once it is broken down, then cell lysate that you get, that is centrifuged at 800 G. And what happens, the supernatant that you get, that supernatant is collected, and it is again centrifuged at higher speed. So what happens that at each rounds of centrifugation, the RPM is increased. The RPM is increased, and the duration is also so with increase in the revolutions per minute of the centrifuge, the what will happen? The heavier particles will be sedimented at higher RPM, while the lighter particles, they will sediment at lower RPM. Okay, so that is what is shown in this figure. So you centrifuge at 800 G, and the pellet that you get will contain the nucleus, will contain the nuclear fraction, and then the supernatant that you get, that is again collected and again centrifuged. And once it is centrifuged in the pellet, now you will get mitochondria, lysosome, and peroxisome. Now you can isolate this pellet fraction, and then again centrifuge the supernatant. And once you centrifuge the supernatant, now these endoplasmic reticulum and plasma membrane, they have less density. Because they have less density, you have to increase the centrifugal force. That is, you have to increase the centrifugal force of the centrifuge of the rotor. So 200,000 G. You have to, so gradually what you are doing, gradually you are increasing the centrifugal force, so that the particles which are lighter in weight or which have a lower density, they settle down. And at each step what you do is you is isolate, or you take out the pellet fraction, and then further you can purify this pellet fraction and isolate the nuclei. Or if you want the mitochondria, you can isolate the mitochondria or lysosome. So this is called differential centrifugation. So differential centrifugation, as the name suggests, different means you are separating different components of the cells on the basis of their size and density. Okay, and homogenize cells. So first you homogenize the cells by different means you have learnt in these lectures. There are different means of homogenization. You can use ultrasonication, you can use a French press, or you can use different chemical methods, enzymatic methods, like adding lysozyme, or you can add SDS by different detergents. So that will also homogenize the cell. And then you ultrafiltrate samples at greater than 100,000 RPM. Okay, so this is in RPM, this is in G. Okay, do not get confused. And so first when you centrifuge it, you get the nuclear fraction. Then when you centrifuge it, you get mitochondria, lysosome, peroxisome. So you remember the order. Okay, so this will be helpful in solving these type of questions. So anyone wants to try this question? So in a certain study performed by, so this is the last slide. I will end my I will end today's session after this. So, in a certain study performed by a group of students on an unknown cell type yielded the following fractions A, B, C, and D on four steps of centrifugation at 600 G, 10,000 G, 100,000 G, and 32 320,000 G. Okay, the fraction A is having the highest mass. So you have been given the information that fraction A has the highest mass. So the fraction which will have the, yeah, any answer? Option number B. Okay, so we will see just now. Let's discuss. Anyone, any other answer? C. Okay, C. So did you read the question carefully? So now, so let's discuss. Okay, so fraction A has the highest mass. Okay, so the highest mass, the fraction which will have the highest mass, will settle down quicker. So it will settle down at lower RPM or lower centrifugal force. Okay, so which is the organelle which is having the highest mass? You see this figure. So you see nuclei. So first of all, the nuclei gets sedimented at 800 G. So at 800 G, first of all, the nuclei gets sedimented because it has a very high density. Okay, so either it can be option B or it can be option C. Okay, because you have to arrange it on the order of A, B, C, and D. Now let's move on to the further information. B being the fraction with the lowest mass. So which is the lowest mass? Is it the ribosome or is it the plasma membrane? So you refer this figure that I have given in this slide. So you see in the at the in the end, what is getting sedimented? Ribosome. Ribosome is that particle which has the least density. So it requires the maximum the centrifugal force for sedimentation. So the lightest particle is B, right? So what will be the answer? Option B or option C? So option B is the correct answer. You had first you had answered the right, given the right answer. Okay, so just just remember that A is the highest density is nucleus, and the lightest is what? Ribosome. Okay, this information you need to have. Nucleus has the highest density, so it will sediment at lower RPM, and ribosome has the lowest density, so it will require a higher density or higher RPM for sedimentation. Okay, option number B is correct. Okay, so you just need to remember this figure. You go through this figure and you will understand the concept very nicely. Okay, so that was all for today. I think we can skip this question. Use of detergents in the process of centrifugal differential centrifugation of cells is uh, so there are different methods of homogenization, right? Chemical method, physical method. All these were covered in your lecture. In the lecture videos, these were covered: chemical methods, physical methods, enzymatic method, right? Mechanical methods. So in the chemical method, one way of homogenizing the sample is by using a detergent which is called sodium dodecyl sulfate. Okay. So sodium dodecyl sulfate, what it does, it lyses the plasma membrane. Because this is a detergent, it will lyse the plasma membrane and it will extract the cellular organelles. So that is the function of SDS or detergents which are used in differential centrifugation. So answer is option C or option number 3. 3. Option 3. That, this one, 26. Yeah. If you have any. No, option B. Option B. Why option B? Did you understand? So you read the question. So here in the question, it is said that the fraction A is having the highest mass. So which is the organelle which has the highest mass in a cell? Nucleus. Okay. So you can eliminate option number A and D. Okay. And now move further and read the remaining sentence, that B being the fraction with the lowest mass. So which is the organelle which has the lowest mass? Yeah, it is ribosome. So B will be what? B will be ribosome. Okay. So now is it clear? So if you see this figure also, you can see that at 200,000 G, at 200,000 G, my ribosomes are getting sedimented. Okay, so that is why the lightest, lowest mass will be of ribosome. Okay, and then fraction. So you do not need to read the further question. Just using this information, you can answer the question. You can read further also. Fraction C and D have intermediary masses with C greater than D. So now you have mitochondria and plasma membrane, which is having higher mass. Is it mitochondria or is it plasma membrane? Mitochondria, right? Mitochondria has higher mass, so C will be mitochondria and D will be plasma membrane. You see this figure, that mitochondria has higher mass. That is why it is getting sedimented at 15,000 G. Okay, while this plasma membrane, you see it is getting sedimented at 100,000 G. Okay, now is it clear? Okay, okay, thank you for your patient listening. It was very nice to teach you all. I hope I hope you are all understood the concepts. Yeah, is there any doubt, or if there are any doubts, we can take them in the next class. Okay, okay, thank you everyone.